Hello, fellow foodies, and welcome back. This is Dr. Cassandra Quave, and you're listening to Foodie Pharmacology, the science podcast for the food curious. Okay, so today on the show, we're going to take a look back in history to the point at which people were making medicines from all sorts of different ingredients. And we're going to be speaking with a modern day scientist who takes a critical look at those ancient formulations and what she calls ancient biotics. So let me introduce you to our guest. We have Dr. Freya Harrison on the line. She is a microbiologist and a professor that works at the School of Life Sciences at the University of Warwick. She researches how dangerous bacteria respond to the environment in an infection. She also looks at how these microbes interact with each other and how they tolerate high doses of antibiotics. She also works on these slimy, multi-celled aggregates of microbes that are called biofilms that are formed during chronic disease. What really drew me to Dr. Harrison's work is this focus on ancient biotics and this reconstruction of historical infection remedies and how she kind of teases them apart and puts them back together and figures out, do they work? How do they work? Why do they work? All those exciting questions. So welcome to the show, Freya. I'm so happy to have you on today.
It's a pleasure. Thank you for inviting me, Cassandra.
Yeah. So maybe we can start by telling us a bit about what are ancient biotics? Where does this term come from and what does that refer to?
So, this is a term that a group of us coined when I was working at the University of Nottingham in Steve Diggle's lab. And we basically made nerd friends with some of our colleagues over at the Centre for the Study of the Viking Age, especially Christina Lee, who works on the history of disease, disability, and death. And through a long story of nerdy copy times and my interest in early medieval history and Steve Diggle's interest in the history of the plague, we thought, why don't we pool our expertise and think about, has anyone actually tried making some of the historical remedies from early medieval manuscripts and testing them in the lab to see if they actually work? And we thought, you know, it's this weird kind of dark spot in ethnopharmacology that people have looked all the way around the world, but not so much in sort of Northwestern Europe and what was happening here before the sort of the scientific enlightenment. It's almost as though we've discarded that period of our own history as sort of superstition and placebo while taking perhaps approaches from other parts of the world more seriously. And while a lot of the remedies and sort of treatments from this part of history have been written about in, beautiful kind of historiographies and people studying their cultural context, there really hadn't been a lot of work at trying to make some of these. So honestly, we thought this would be fun. We thought we would have a little side project, an excuse to do some cross-disciplinary work. We might get a little paper. And here I am 12 years later. Um, uh, and we've just submitted a patent application for an antimicrobial formulation inspired by, developed from what was used in one of these remedies. As you well know, I've now graduated 2 PhD students who've studied some of these remedies because you kindly came and were their examiner. Um, and it's really just mushroomed. And so yeah, we decided early on that a nice snappy title for this was Ancient-biotics. Um, and please don't write in. We are well aware that to historians, ancient is a little older than medieval, but sounds a bit like antibiotics, and it's nice and memorable.
I like it. I mean, we need some creative ways of looking for new solutions when it comes to antimicrobial discovery. As a point of reference for the listeners, we are in or entering into what may be known as the post antibiotic era. A lot of our existing classes of antibiotics don't work as well as they once did or are rapidly losing efficacy because of the rise of antimicrobial resistance. And we desperately need to fill that pipeline. Now, when I think about the logistics of, how do you go about looking to like a medieval remedy? I know that some of your work started with this book. It was called Bald's Leech Book. Is
Mm,
that right? So
that's right.
how is this written in like medieval English? How hard is that to interpret? I'd imagine it's a bit different than modern day English, right?
So it's written in Old English, and this is, it is the language which modern English developed from, but it is a different language. It has a different grammar. So you do need there needs to be an expert in that area reading this and translating it. It's not something a scientist with a dictionary can do. Um, and that person has to be able to translate the cultural context as well. If the person who wrote this book, so Bal, the physician, or the leech, or their scribe, what did they mean when they said this remedy is for a black blane or for a wound that is hot? What did they mean by a worm, for instance? Um, so this is really something that has to be a collaboration between scientists and the arts and humanities. So we had a huge discussion, for instance, that first remedy we looked at, which was a salve for a lump in the eye. It mentions two plant species. One we know unequivocally is garlic. People have done studies of plant names. There's illustrations in herbaria. We know that word, garlic. There was a second plant species, which is clearly another allium, which is called cropleek. We don't know what that modern plant is. We know it's an allium. So we had all sorts of discussions about, is it an onion? Is it a leek? What were medieval onions like? How much have we bred these vegetables? What candidate vegetables should we try? So yeah, there's an awful lot of interpretation. And I think, it is important to say, we would not say we have reconstructed exactly what a medieval physician would have made. We're doing our best with the resources available.
Now that's such a great point because you're dealing with a different language. The terms for these ingredients are different
Mm,
today. And the plants that they're talking about may not actually exist in
yeah,
their current form because you're right. I mean, we know that crop genetic diversity changes over time. And you're also mentioning plants that could be considered foods, garlic, leek. If I remember correctly, was wine one of the other ingredients to this?
it was. And that's a great example because as I understand it, virtually all modern vines in Europe have been crossbred with a North American species or variety. So aside from some particular heritage variety somewhere in the south of France, we don't have the same variety of grapes that people in the medieval period would have made. So we then thought, well, is the variety important? There's some amazing work on grapeseed deposits in Roman archaeological strata and early medieval archaeological strata where people have worked out the closest modern varieties. But we also thought, is it the terroir of the grape? Is it actually the geology of the soil that might determine what compounds are present within the wine? So we this kind of mushroomed in all sorts of directions. It is, as a microbiologist, this was a very, very different approach than saying we order some tetracycline from Sigma Aldrich and we make it to this concentration.
No, absolutely. I mean, I think a lot of our modern dogma in science, I'll use the word dogma because I think we really do have kind of this very dogmatic ascription to certain steps in science. It's very much like you take one molecule and you try to test it
Mm,
against one target. And that's what a lot of our modern day science is based on. Because when you start bringing not only different ingredients, well, if you're bringing different molecules together, that makes things more complicated with polypharmacology. But you're not just bringing more molecules together, you're bringing whole other, natural products from different living organisms, which really constitutes thousands of new molecules coming together.
absolutely.
Yeah.
Absolutely. And I can show you an example of this sort
Oh,
of
yes.
the eyesalve. So I have this here. This was the remedy we first made, the eyesalve, and it is a mix of garlic, the second allium. We tried onion and leek, wine, and cow's bile. So if you were to do liquid chromatography mass spec on this and work out what compounds are in it, we've done it. We tried. There's several hundred individual compounds in here. Um, and this is kind of a callback to the episode you had with Nadja Chek, where she was talking about the importance of synergy in the activity of plant extracts. And that was a beautiful explanation. And you're exactly right. We not only have multiple compounds from each ingredient, but then how they act together.
Now that's incredible. So you have this concoction, and for the listeners that aren't watching the visual version, I'll just kind of describe it. It's a brown liquid in a vial. So it's this brown liquid, and then apparently they were adding this to the eye. Are they talking about like styes, like when you get an infection in your eyelid? Is that what the thought was from the historical interpretation?
That's what we think they meant. So, um, what they said was you take these ingredients, and they were kind of specific about the amounts. They said equal amounts of garlic and the second allium species, pound them together, um, and then mix them with equal quantities of wine and bovine bile. Then you put them in a brass or a bronze pot for 9 days. At the end of the 9 days, you strain out the lumps through a cloth, and that liquid you drop into the person's eye using a feather as a dropper.
Oh,
Um, and the word they use for the lump in the eye is a wen, which is an old English word that just means a lump. And we thought, well, there's various reasons you could get a lump in your eye, but one of the most common ones would be a sty. And if you look at those ingredients, that makes sense. All of those ingredients we know as microbiologists have some antibacterial activity, at least in a test tube. So garlic and other alliums are just some of the most amazing chemical factories in the plant kingdom when it comes to sort of, reactive molecules, things with interesting sulfur groups that you read up chemistry, things that affect microbes. Then you think, wine, it's alcohol, which can be antimicrobial. There's potentially really interesting things coming in from the grapes. So, the sort of, all the different organic acids, polyphenols, resveratrol in there that we know have effects. And then the bile sounds gross, but one of the roles of bile in your diet is to break down fats. And so outer membranes of bacteria are basically made of lipids. So bile punches big holes in those membranes and kills them.
oh
So when
wow.
you look at, and then you've got the copper as well. So we then thought, okay, you're also potentially leaching copper salts from this brass or bronze vessel. And as most people know, copper's wonderfully antimicrobial. Our own immune systems, some of our white cells keep little reserves of copper in pockets inside them to kill microbes. So we thought, looking at all these ingredients, this does look like something that could kill bugs, and that may have been used to kill bugs.
Now that's amazing. I think what really caught my attention from the early work you did with Steve Jeggle too is that you guys actually broke this recipe apart. So you said, well, is there an antimicrobial activity we take part this part or that part, or
Mm,
is the copper pot necessary? And that was just the most fascinating study because it really, if I remember correctly, it really was the whole formulation that was most effective. So like
it
the ancient
was.
remedy in its whole was best.
It
Yeah.
was, and this we were really shocked. Honestly, we thought this might have some activity because most things do in a test tube. We initially had the hunch that we were basically going to be making an acidic solution that leached copper salts out, and the copper would kill the bacteria. That wasn't what happened at all. We actually found the only thing you didn't need was the copper pots, and if you mix these ingredients together, they absolutely nuke these preformed biofilms of the bacteria that cause styes. And you're right, we tried making versions where we missed one ingredient out. They didn't work. We tried using each ingredient alone, didn't work on its own, or not to the same extent. And that's kind of interesting because people have looked a lot at garlic in particular as a potential antimicrobial. Some of the compounds in garlic do really interesting things to bacteria. They can kill them, they can mess with how the bacteria signal to each other to become virulent inside a host. And so there's been a lot of research activity on that, but nothing has translated from this looks great in the lab to something that actually works in a patient. So to be able to say, actually, when you take this ingredient and you add these other three things, you get something that is much greater than the sum of its parts and might be worth looking into. That was fascinating. And it's, if people who are listening haven't listened to Nadia Chek's episode, that's, she explained this idea of synergy, of having multiple compounds attacking the bacteria on multiple fronts at the same time beautifully.
No, I love that. Yeah, I mean, if you look at the history of traditional medicine, so
Mm,
many of our systems of traditional medicine use these multi-component polyherbal formulations to really hit at the target from multiple ways. I mean, as I mentioned at the top of the show, antibiotic resistance is such a challenge. I mean, we have our current projections are that we may reach up to 10 million deaths a year by 2050. I mean, we're not that far away from that. And we need new solutions. One of the
Yeah.
contributors, of course, to antibiotic resistance is not just resistance to the antibiotics themselves, but also the kind of, through genetic mutations, but also the physical barriers, which you mentioned. And I think we can also pull up an image of that, of what a
Yeah.
biofilm looks like. It's this slimy matrix, if you can imagine, like maybe a rock in a stream with these slime on it. Tell us more about how, and we're showing an image of this now for those of you watching the video version of the episode. It's a group of bacterial cells that are kind of globbed together in this polysaccharide matrix, kind of sugars that are made and DNA and all this other goop. What makes biofilm associated infections so challenging to treat?
So it's kind of a multi-hit effect, really. So firstly, there's this physical barrier. The bacteria, they secrete this slimy mix of these sugars and proteins, and they can co-opt proteins from the host into that as well, um, or other molecules from the host to make this overcoat. And if you imagine you've got this very hydrated network of molecules which have electrical charges on them, that's going to mean that molecules that are too big or that have the matching charge can't get through. They can't penetrate that matrix and they can't actually reach the bacteria inside. So you can get this almost kind of like steric hindrance where you're trying to throw drugs at a biofilm and it's not actually getting to the bacteria at all. But when bacteria switch to this lifestyle, they actually change an awful lot of their metabolism as well and a lot of their physiology. So when bacteria make this switch to create a biofilm, they will also upregulate antibiotic efflux pumps. They might change the structure of their membrane slightly so that it's harder for drugs to cross it. Sometimes in a biofilm, especially in these sort of deep layers of a biofilm, and these can be hundreds and hundreds of bacteria deep, there's very little oxygen, there's not many nutrients. So the bacteria, they don't die, but they stop metabolizing. They just become very quiet, very quiescent. And that means that they're not making any of the targets that antibiotics usually hit. So if you have something, an antimicrobial that targets protein translation, and your cells aren't translating anything into protein, it's not going to affect them. If you have something like a penicillin that targets the cell wall and how it's being made, if a bacterium is not dividing, it's not trying to make new cell walls, so the penicillin is not going to get it. Um, and the other great thing, or great thing from the microbes' point of view, is that these are diverse communities of bacteria where you can have division of labor. So if you imagine a biofilm of different bacteria, and perhaps you've got some bacteria that are making enzymes that break down antibiotics, you can be a neighbor of that bacterium and not have to make your own enzyme, be protected by your neighbors who are detoxifying that environment. And these biofilms, they're really mainly thought of in the context of long-lived chronic infection. Um, Something 70 to 80% of chronic infections, things that last for months and months and don't respond to antibiotics, are biofilms. So these are things like what you might see in a diabetic foot ulcer, in the lungs of people with cystic fibrosis, or the example you showed, that was an image we took in our lab of Pseudomonas bacteria forming a biofilm on an endotracheal tube. So that's the tube that would connect you to a hospital ventilator if you were on mechanical ventilation. Um, and if you want to see particularly unpleasant examples of biofilms, if you Google for images of these tubes when they've just been removed from a patient, you can physically see the gunk and the slime stuck to them.
Yeah, it's so challenging. I think implanted medical devices, anytime you have tubing coming into your body, whether it's a urinary tract catheter or a port, or patients undergoing cancer therapy have to have often ports embedded for delivery of their cancer medications,
Yeah.
intubations in the ICU. These are all spots, hot spots of opportunity. You know, medical implanted devices like hip replacements, joint replacements, knee replacements, cardiac devices, all of those are very susceptible to biofilm formation. So it's a weak point, I feel like, in our area,
Mm.
of or arsenal of antibiotics to treat these kind of very difficult recalcitrant infections. So when you're looking back at this ancient biotic formulation, tell us more about some of the observations that you all had. So you mentioned antimicrobial or antibacterial
Mm.
properties, but how did this old remedy, and this is this like 1,000 years old? Was there yeah.
Yeah, we think so. The book it was in, Bald's Leech book, and a leech book is a medical book. Leech is the Old English word for doctor. We think it was written, I say we, the historians think it was written around about the year 1000, potentially in Winchester, which is a city in the south of England.
Wow.
And this remedy doesn't have an exact parallel anywhere else in medical books of the period that survive or in classical medicine. There's some similar things. There's kind of some remedies that have two of the same ingredients, but nothing absolutely identical to this. So we think this was potentially a homegrown English remedy about 1,000 years ago.
Wow. So 1,000-year-old remedy that you're now studying to treat modern day,
Mhm.
incredibly difficult types of infections. So tell us more about how this works in a biofilm context. Does it punch through that slimy matrix at all? Like what are you finding in the lab?
Yes, and one of the most exciting things was that it could kill bacteria in an established biofilm and it could penetrate that slimy matrix. And we are about to submit a paper which describes the mechanism of action. And I really want to name check the two PhD students before I tell you this. So the first is Jessica Fernapado. And during her PhD, she worked on the mechanism of the eyesalve and gained some insights into that. She also worked out that she could make a sort of semi-synthetic version of this, and that actually she could take one of the ingredients, one compound from another ingredient, and get most of the activity. So we think when you mix the original four, there's some extraction, some chemistry happening, and actually we can recapitulate it a little more simply. And then Oluwatosin Orobaba, who has just finished his PhD, um, has taken this further and he has worked out in detail how this original four ingredient formulation attacks bacteria. And coming back to that idea of synergy and having multiple compounds, this really batters the bacteria on multiple fronts. Um, so it it damages the outside of the bacteria, their membranes. So it makes their membranes more permeable. So things can leak out, things can get in. They can't maintain that kind of organismal integrity. And this is really interesting because we do have antibiotics that affect membranes in this way. So colistin for listeners who are familiar with antibiotics would be something that affects membranes. Um, and this is really important because actually membranes are really, really evolutionarily conserved. Bacteria, you can't mutate your membrane very much and still survive. So it's hard to get resistant to a drug that attacks the membrane. But it doesn't just do that, or eyesalve. What it also does, um, it can interfere with quorum sensing, and that's the cell to cell communication between bacteria. And they use this kind of communication to tell each other to stop building a biofilm or to switch on the expression of proteases that degrade host tissues. And it can interfere with quorum sensing in some of the bacteria that we looked at. Um, it also, once it gets inside the bacteria, it can mess with some of their central metabolism as well. So Tosin has shown that it stops some of the pathways inside the bacteria that make the building blocks of nucleic acids. So if the bacteria is trying to make more DNA or RNA, that process is going to be affected when it's treated with this eye cell. And we tried this. There's kind of two big divisions of bacteria. There's gram-positive bacteria, gram-negative bacteria, and they have slightly different membranes and surfaces. And we showed that there were some commonalities in how this attacks the two types of bacteria, but some differences as well. And we could really see those membrane effects when we looked under the electron microscope again, I say we, when Jess and Tosin looked into the electron microscope, they could really see this kind of damage to the membrane. So you could see deformations and sort of wrinkling and blebbing of that membrane.
That's amazing. I mean, this is what we need when it comes to antibiotics, I feel, because again, single molecule, single target, it's just so easy for microbes to develop resistance to that. It takes one mutation. And for those in the audience that aren't familiar with the life cycle of microbes, there are many really dangerous microbes, bacteria that can basically double their population size, every 15 to 30 minutes. So they're constantly reproducing. You've got exchange of genetic information within these populations as well. That just there's so many shots on goal to find a way to get around a
Mm,
drug. And so when you're hitting them from all angles, it just really makes it a lot more challenging for them. So it's an evolutionary race, I guess. And I love that we might have
absolutely.
1,000-year-old remedy that can help us a bit in this evolutionary race. Well, in addition to your work on Bald's eye salve, I know you've also explored some other remedies. Maybe we can talk a bit about your work with Erin Connolly on vinegar and honey. And yeah, tell me more about that and kind of how that connects to this ongoing focus area.
Yeah, so Erin and I have been sort of collaborating since those early days of the Ancient-biotics project, and in the last few years, we've been working on this honey and vinegar project. And um, Erin's a really interesting person. So she has a first degree in biomedical science, but then she did a master's in medieval and early modern manuscripts, alchemical manuscripts in particular, and switched fields. So she ended up doing a PhD on a particular edition translation of a historical medical book called The Lily of Medicines. So she has this amazing dual background. And one of the many things we got talking about was this really common use of honey and vinegar. And you find it an awful lot in remedies that are designed to treat what appear to be symptoms of infection. So, wounds that are purulent or hot or red or yellow, um, things like eye infections, pustules or sores in the eyes or the mouth. And there's this really big pattern wherever you look in the world, wherever you look in history, honey and vinegar being put together to treat these. And when you think about this again from a modern perspective, the use of medical grade honey to manage wound healing, where you see biofilm infections, is now a standard of Western medicine. You know, people know that there are these multiple properties of honey that can make it antimicrobial and provide a really good environment for wound healing. And when you think about the vinegar, the main acid in vinegar, acetic acid, that's actually a standard treatment for antibiotic resistant burn wound infections, particularly those infected with Pseudomonas. And acetic acid's quite interesting because it can kill bacteria at very, very low concentrations, way below the concentration you'd find in table vinegar and way below the concentration where it's simply you're putting a really low pH solution on something. And so we wondered, have people combined these in the modern world? And we found that really no one's looked at combining acetic acid and medical honey. We had a fantastic master's student, Anissa, who did systematic review for us. She found one single paper where people had done something approaching looking at putting honey and vinegar together, but they hadn't designed the experiment in a way that allowed you to test for whether you got a synergistic effect on the bacteria. And by that I mean is, what I mean is, do you kill more bacteria than you'd expect by just adding the numbers of bacteria you kill by the honey alone and the vinegar or the acetic acid alone? Or do you get more bang for your buck?
Yeah.
And then
That's
we
a
spoke, sorry.
that's a oh no, just I just want to reiterate. Yeah, it's like that's the basis of synergy. Synergy is a
Mm,
world where 1 plus 1 does not equal 2, it actually equals 4 or 5, right? So it's like it's not just an additive effect. So that's okay. Sorry.
exactly.
Yes.
Yeah, perfect. Um, and then we spoke to some clinicians. So we spoke to a reconstructive surgeon and wound care nurses. Just, do you ever put the the medical grade honey ointment on and then put an acetic acid dressing over the top. And no one did. No one had heard of this. We couldn't find any records. So we thought, right, we have to try this. And we use similar ways of growing biofilms in the lab as we did for the eyesalve. So we can make little synthetic wounds out of collagen and synthetic wound fluid in the lab and grow our bacteria in them. So they form these sticky biofilms. And then we can add on just the honey, just the vinegar or the acetic acid or both of them. And then we can count how many we kill. And what we found is that you can get synergy. So we found with some medical honeys and some vinegars, or with some medical honeys and acetic acid, we were getting these big synergistic effects on biofilm killing, where we were able to take a dose of honey that on its own was subbactericidal, and a dose of acetic acid that on its own was subbactericidal, put them together and get a lot of biofilm killing. We also found some really interesting things with the vinegars. So this was a really fun project because we had to think, okay, there's lots of different types of vinegar. What were people using in different periods in history and around the world? Maybe here in what is now the UK, people were leaving wine or mead to go
Mhm.
shop and using that, whereas perhaps in the Mediterranean, they were making date or pomegranate vinegar.
Oh,
So we had a fantastic shopping trip. We bought as many different types of vinegar as we could. We tried to go for things that were unpasteurized because we thought, maybe the bacteria in the vinegar and that the fungi doing something as well. And we found this huge variability in the activity of vinegars. And we found there are vinegars that are much, much more bactericidal, than you'd expect just based on their acetic acid content, which again is telling you there are compounds in them that are perhaps synergizing with the acetic acid.
yeah.
Um, yeah,
I'm thinking of like apple cider vinegar. There are lots of kind of homemade
yeah,
remedies using that. And, we all know the flavor. I mean, this is a food show, so we're talking about bacteria, but we know the difference between balsamic and red wine vinegar, white vinegar, apple cider. I've never had pomegranate vinegar. I bet it's delicious. Rice
yeah,
vinegar. I mean, and they all come in different colors and flavors, and those colors and flavors are because of the different plant chemistry. So you've got polyphenols and tannins and all kinds of different things I'm sure that are present. So tell us more. Like, what did you find? Like, were there certain vinegars that performed better than others?
there were, and we've published this paper, and we found that pomegranate vinegars were just amazingly bactericidal. And again, we sort of dug into the literature, and people before us had taken that one molecule approach, and they'd looked at particular molecules that are present in pomegranates and shown that they had some antimicrobial effects. So we thought, well, maybe these things actually synergize with the acids when you've made a vinegar. So we then went back and we got pomegranate juice and thought, well, if we add acetic acid to pomegranate juice, do we get synergism? And we do. So I think there's this gold mine of something that could be in pomegranate juices and pomegranate vinegars that is still to be explored.
Oh, that's
Um,
that's exciting. We published a paper in 2025. There's a PhD student I mentored from University of Sassari in Sardinia, where you're in Europe, so maybe you get a chance to travel. I love traveling in med, but Sardinia in particular was a beautiful place to go and visit because just gorgeous food, people, landscapes, amazing place. But they have these cultivated like plantations of different varieties of pomegranate, and they were looking at pomegranate, the peel of the pomegranate. And she found actually an interesting antimicrobial against staph from the pomegranate rind. It's called a punica galen.
yeah,
Yeah, is the name of it. So, but microbes also transform the composition of plant secondary metabolites. So it could be something you don't even find in the native plant extract, but needs to be transformed by another, like a fungus or whatever's
mm,
or a bacteria that's transforming it from a wine into a vinegar. I guess it's a bacteria that does that transformation, right? I'm trying to remember
I
my microbial transformation chemistry. I think it's, but yeah.
I am having a mental blank, and I think potentially is lactic acid bacteria, but we may have to look that up and come back to.
Yeah, yeah, yeah. No, it's Acetobacter. Acetobacter. It's yeah, Acetobacter acidii. I have a weird memory for Latin names. Don't ask me people's names, but yeah, that's it. So it's a bacteria. But I wonder if there's some sort of metabolism happening there where they're breaking down molecules or yeah, transforming them. That's
Exactly.
fascinating. Yeah,
And are
there's
there
such
even maybe probiotics in there
yeah.
that are helping? That's something we looked at with the eyesalve and we found out it
Mhm.
wasn't. But when you're looking at things like vinegars, perhaps it is. Perhaps there is also something with adding in good bacteria.
Yeah, to kind of help with the microbiome environment in the skin.
Mhm.
I've been to a number of these wound healing meetings. It's I don't have you been to some they're different from microbiologic meetings, like microbiology meetings, because every poster presentation is just these gory images of really purulent wounds, burn
Yeah,
wounds, abscesses. It's really you have to have a solid stomach to walk through that poster hole at the conference. But I've seen a lot of presentations on folks using, Manuka honey as a medical grade honey to help with, really does seem to help patients recovering from these,
yeah.
large scale burn wounds and prevention infection prevention. That's fascinating. So where are you taking this work next? I mean, this could be a really, really interesting area to help patients that are battling these kind of chronic or at risk of these skin infections after wounds.
Absolutely. And I think this is where the kind of the ancient biotics work, diverges into several themes. By looking at how these things are put together, there's some really interesting humanities driven work in looking at knowledge transfer and how these things are
Mhm.
developed. But then from our perspective, we're now thinking like, okay, this is looking promising, but nobody wants to have their doctor say, right, I'm gonna mash up some garlic and some cow bile and in 9 days I'm gonna put it on your ulcer. We need something that is actually reproducible and defined and dosable. Um, so what we've been doing and Jess and now Tosin have been doing is trying to identify exactly what molecules present in this kind of like 300 odd compound mix of bullseye cell are responsible for its antimicrobial activity. And can we use it as a sort of a little database or a foundation for making a multi-component natural product preparation that could be turned into something like a wound dressing? So you want,
Amazing.
and I would not have predicted we would get to this point, you know. Um, but what um Tosin has really led on through his PhD is that funneling down of these compounds. And we've been able to identify basically four molecules. We've been able to play with their concentrations and kind of optimize the concentration and the ratio. And then we also had a bit of a think, polypharmacology is not unique
Yes.
to looking at say at historical microbials, as clinicians do. So we thought, are there any known antimicrobial adjuvants that are in clinical use that we might be able to add to this and get even better synergy or a bigger additive effect? And we tested quite a few sort of antibiotics and substances used in clinics, and we were able to find a fifth thing that we could add to this synthetic cocktail, and again really optimize the concentrations. And so we have now submitted a patent application that describes this formulation. Um, we've repeated a lot of the mechanistic work to say, right, this is what the raw eyesalve did to bacterial cells. Now we've got a defined cocktail. What is it doing to them? And
Fantastic.
so, and it's just been stunning. And we have been able to put that together. So I'm hoping we'll submit that paper very soon.
That's great.
Um, and then just to go back to your point about evolving resistance, that is absolutely crucial. There's no point saying, we've got this amazing thing that kills biofilms if you would put it in a patient and a couple of bacterial generations later, they've become totally resistant to it.
Mhm.
So we've also spent a lot of time trying to evolve resistant bacteria in the lab.
Yeah.
Um, and completely failing.
Yes,
Right.
that's great. Just so background for listeners is like one of the ways that we figure out how antibiotics work is we'll basically feed them small doses of the antibiotic compound you're trying to develop until a mutant arises that's resistant to the drug. Then you can sequence that mutant and understand, okay, this is how it evades. But you're doing this and you're not seeing those mutants arise.
No,
That's exciting. That's really
which
exciting.
it was
Yeah.
kind of doubly exciting and kind of
Yeah.
infuriating because if we had found it, we'd have been able to work out the mechanism a lot easier.
Yeah.
But yeah, we have tried various different ways of kind of trying to ramp up the selection pressure in the lab and try and get bacteria growing in higher and higher concentrations of our cocktails. We've even gone back to some of the when we treat the biofilms in the wound and said, okay, well, some of the bacteria survive. Were they just lucky, or did we have a random mutant? No, they were just lucky. They're not resistant. Um, and we have really been unable to evolve resistance to this in the lab. Now, I would never say any antimicrobial is evolution proof, right? There is no antimicrobial out there to which some bacteria have not evolved resistance, right? There's things that are resistant to bleach out there in
Yeah.
hospitals, right? Um, but what this does at least say is that it's not incredibly easy
Yeah.
for bacteria to. It's not like they can mutate one gene and become resistant.
That's
Um,
incredible.
So that is really, really heartening that we have something that we think really is worth taking into clinical trials. We did a lot of work on the safety profile of the original eyesalve formulation, the garlic onion violent wine. Um, it's not particularly pleasant to have on your skin. It was about as irritant as the glue on a surgical dressing.
Mhm.
Um, but yeah, that is something that we would then need to go into a lot more detail on.
Yeah. Well, I think it's fascinating this trajectory you've taken from, you know, historical curiosities. We didn't get into the also kind of the other fun aspect that you all do, and that's around historical reenactment. So it's not LARPing, so to speak, but it is a historical reenactment of different eras. Doing that with friends to say, I love that. Like you would tell us a little bit more about that too. Just want to make sure we catch that.
Yeah, so this is kind of how I initially got interested in this work ultimately. Um, I guess when I was start of my PhD, I thought it would be fun to learn to do some sword fighting. So
Why not? I love
I
it.
joined a historical reenactment combat group, did that. Fantastic fun. It happened to be an early medieval group, and I thought, this is a period of history I'd not learned about in school, and I got very interested in sort of the craft side of it, making costume. Thought it'd be interesting to do something that shows sort of literacy, which got me into a way to do this would be to look at historical medicine. Um, and yeah, so a lot of my spare time I spend either fighting with swords and daggers, which is a lot of fun and a good stress relief. But then I also end up going to events and living history festivals where I'm portraying an early medieval English doctor, and talking to people about the kind of things people might have used in those times and how they might have thought about illness.
That's amazing. I love that. It's so much fun to bring together your kind of fun hobbies and passions with your, deep microbiological knowledge and to share that with other collaborators. It's fantastic. And you've taken it all the way to, like you mentioned, getting a patent filed. Next steps. I mean, if there are any listeners out there in the wound space, I mean, I'm assuming this will be available for licensing from your university, or are you plan on starting a startup company, or still kind of to be decided. But development takes money. So, those that are interested, like reach out to your university office of tech transfer, I'm assuming. And I wish you the best
Absolutely.
of luck. And, I know this is a food show, and we talked a lot about infection today, but I will say we talked about treatment of infection with food, honey, vinegar,
Yeah.
garlic, leeks, wine. I think there's so much that we can still learn from the past, and it's great to see how you can do that in a really rigorous modern scientific lens. So thanks for sharing that with us, Freya.
Thank you.
Yeah. Yeah. Well, thanks so much for coming on the show. Thank you to our listeners for tuning in. Again, we are in season 8 of Foodie Pharmacology. I want to give a big shout out of thanks to our producers, so Rob Cohen and Christine Roth, that help us keep the show going. For those of you listening on audio, keep it up. If you're following us on Apple Podcasts, be sure you give us a 5-star rating. Leave a comment, share it with your friends. We can always use the help to get the word out. If you really love the show, buy me a coffee. I can use some extra caffeine. We use the Buy Me a Coffee portal on our website. You can find it at foodiepharmacology.com. That helps us really help cover some of the production costs. You can also watch the full video version of this on our YouTube channel at Teach Botany. So thanks so much for listening. Stay healthy out there, and we'll see you next time.